The question we will answer today is how can we treat prostate cancer? This presentation will introduce a new proposed drug treatment against prostate cancer, one of the leading cancer types diagnosed in men. It is estimated that 16,600 men will be diagnosed with prostate cancer in Australia in 2017. Prostate cancer is a result of abnormal cell growth in the prostate due to both inherited or acquired mutations. These cells continue to multiply and can leave the prostate and reach local or distant sites in the body. In Australia, prostate cancer is the most commonly diagnosed cancer in men, and sadly more than 3,000 men die of prostate cancer every year. Our proposed treatment will target metastasis. This is one important and severe part of cancer, especially prostate cancer, since it allows cancer cells to leave their local area and spread throughout the whole body. Now we will look at MMP secretion as a prostate-specific target. What are MMPs? MMPs are secreted endopeptidases involved in the degradation of the extracellular matrix. MMPs break down collagen and fibrin, which promotes tumour invasion and migration into the surrounding tissue. Secretion of MMPs is important in many hallmarks of cancer, such as metastasis, proliferation, and angiogenesis. So how does this affect prostate cancer? Specifically, MMP2 is overexpressed in PCA tissues, and its secretion was significantly associated with a grade of PCA malignancy. MMP2 upregulation is associated with high progression and poor prognosis. So how will we inhibit MMP2 secretion? Cannabidiol is a non-psychotropic cannabinoid compound and binds to CBD1 and 2 receptors, causing inhibition of MMP2 secretion. CBD-induced inhibition of MMP2 expression is selective, as levels of other MMP family members were unaffected. This decrease in MMP2 secretion downregulates the ability for cell proliferation, metastasis, and invasion, and thus decreases the invasive cancer phenotype.
To target the CBD to prostate cancer cells, we will use an antibody-drug conjugate. This form of treatment combines monoclonal antibodies, which target a specific antigen, attached to a biologically active drug by chemical linkers. The human IgG antibody will target the prostate-specific G protein-coupled receptor. PSGR is exclusively found on prostate cells, which makes it a perfect drug target. In addition, during prostate cancer, the mRNA for PSGR is upregulated, and with the help of chaperone proteins, there is an increase in the receptor expression on the cell membrane. Hence, having an antibody binding to this site will increase the specificity of CBD to the cancerous prostate tissue. Once bound to the PSGR antigen, the antibody and attached drug will be internalized into the cell and the link degraded, so the CBD can bind to the cannabinoid 1 and 2 receptors and downregulate MMP2 secretion, thus reducing the degradation of extracellular matrix and metastasis. As well as allowing drug specificity, the antibody will enhance the body's innate immune attack on cancerous cells by activating the complement pathway and antibody-dependent cell-mediated cytotoxicity. However, there are some limitations to the proposed treatment. With the use of antibodies, there is always a chance of non-specific antigen binding or binding to PSGR on non-cancerous prostate cells. But since CBD is non-toxic, off-target delivery would not be detrimental. Additionally, the PSGR may not be present on all cancerous prostate cells, so MMP secretion will remain uninhibited in these cells. In terms of drug safety, oral CBD doesn't present opioid interference, thus is safe when taking painkillers, and it does not display signs of abuse liability. Commonly reported side effects are tiredness and changes of appetite and weight. CBD also acts as a cheaper alternative to chemically derived MMP inhibitors such as batamostat, a broad-spectrum MMP inhibitor which costs $12,000 for 300 milligrams. In contrast, a 10 ml bottle of hemp oil containing 300 milligrams of CBD costs only $49. CBD antibody drug conjugation is an effective form of drug delivery to prostate cancer cells. Through the action of MMP2 inhibition, cancer metastasis should be decreased. This treatment has great potential for further research and development due to its importance in targeting a major hallmark of prostate cancer in a specific, controlled and cost-effective manner. Thank you.